Hi everyone, my name is Kelly and I wanted to show you a quick activity you can do at home with materials you have in your kitchen that can help you understand a bit more about a really cool structure in a cell, and that's the cell membrane. So this video is going to be really helpful for you if you're a grade 8 student and you're thinking about the structures and components of a cell, but it's also really fun if you're just interested in understanding how models work, or if you're just interested in understanding a little bit more about a cell membrane. Okay, so just really quickly, the cell membrane is the outside layer of a cell. It's made up of fats called phospholipids, and it also has things in it like proteins, and it controls what comes in and out of a cell. So you can imagine that a cell, a human cell or an animal cell or a plant cell, all need to take in nutrients and things to make it work, like oxygen or glucose, but it also needs to be able to get rid of things that it doesn't need anymore, like waste products like carbon dioxide. So the cell membrane functions to allow things to come into the cell as well as to let things leave, among a bunch of other functions. But as you can imagine, because it's part of us and animals and plants and everything in the living world, it's fairly complicated. And so what scientists like to do all the time is to take a complicated system and find a model that's a bit more simple so that we can understand things about that complicated system. Okay, so I want to show you this simple model. So to look at a cell membrane, we can model it using a soap film. So essentially today what we're going to do is make a big bubble mixture, and we're going to look at the properties of this bubble mixture. So what you need, and I'll tell you the ingredients and then you can pause the video and go get them. What you need is some warm water, you need some sugar, so about 2 cups of warm water, you need about 2 tablespoons or 1/6 of a cup of just granulated sugar, and then you need about 50 milliliters or about 1/4 of a cup of just dish soap. You'll also need some straws. These are silicone straws, but you could use anything you have in your house that's cylindrical. You need some rope, some string, then you need a dish that you're going to be able to make the bubble mixture in, and then you need a little bit of just plain water, and then any other fluids you have around your kitchen that you might want to test. So here I have oil, just cooking oil, and here I have apple cider vinegar. If you had alcohol, like cleaning alcohol, you could use that. You could use any type of vinegar. I'm trying to think of other liquids. Really, like anything. You could take juice, you could take milk, anything that you want to test. And that'll make a bit more sense as we go through this. Okay, so we have the warm water, we have our soap, we have our sugar. So let's make our bubble solution first. So first I'll take the 2 tablespoons of sugar and I'll just pour it into the warm water. It's important to have warm water because it'll make dissolving this sugar a little bit easier. And then we'll just mix up that sugar in the warm water just until it dissolves. So this is kind of like a baking experiment. Okay, now the next step is just to put in the soap into your water. Not a huge deal, but you want to try to pour it in slowly and mix it around slowly because we don't want to make a huge amount of bubbles on the surface just yet. Okay, that's pretty good. And then just mix it in so we have a full bubble liquid. You see I'm not making too many bubbles at the surface. Okay, oh, a few. Okay, and now I'm just going to pour it into our container. And again, try not to make too many bubbles. It's kind of hard. Okay, so there's that. And now we're going to want to make a frame so we can actually see a bubble film. So I'm going to take my string and pass it through the first of the straws. And you can speed this along if you see where I'm going, but I just wanted to show you how it works. Okay, and then pass it through the other one. Okay, sorry it's taking so long. Okay, and now you want to just tie that off. So you want to make sure that this frame we're making is going to fit in your container with the bubble solution. So we don't want it to be too big. So I'll just quickly tie this off and I'll show you when I'm done. It's gonna kind of form like a little rectangle. Here's our knot. Okay, and so you see I've made like a little rectangle with the string, and you see I've left some string over here. So what I'm gonna do is I'm gonna make like a little holder. So I'm gonna pass it back through this end of the straw and tie it off, and it'll make a little triangle holder, okay? Okay, so passing the string through, almost there. This is the slowest part of the exercise. Okay, so now I'm just gonna see how I've kind of made that little holder. Okay, so I'm just gonna tie this off at the same place that I already made a knot. Okay, and so you can see what I have here is a triangle kind of at the top of a rectangle with these straws. I'm just gonna hide that knot. Oh, I guess I tied it in the wrong place, but it'll work anyway. It just won't be as pretty. Okay, so now what we're gonna do, and you can kind of imagine it, is we have a frame, we have a bubble mixture in here. We're gonna just create a bubble film in this frame. So you just dip it in and then lift it up. And can you see that kind of sheen? There's a film actually in here. Yeah, there you can see it. Okay, so if it doesn't come out right away, just dip it a few times until you can get a film. Now go ahead and pause the video now and make up your bubble solution and make your frame, and then join me back when you're done. Okay. Okay, so you've made your frame, you have your bubble mixture, you have a little film. Now what the heck are we gonna do? Okay, we're going to think about this film as representing a cell membrane, okay? So this film is just made up of soap and water molecules. What are some of the things we want to think about when we're thinking about things that can come in and out of the cell? So we might want to understand how you could go from one side of a membrane or one side of a film to the other without, say, breaking that film. So let's just play around with this. How can we do that? So we have a finger, hopefully you have a clean, dry one. Why don't you go ahead and see if you can put your finger through the film. Okay, I'll give you a second to do that. Okay, now you've done that, you might have predicted what might happen, and then you observed what happened. So I will just go through that exercise with you. I'm gonna make a prediction. I think that my understanding of bubble films, if I try to touch it, it's gonna pop. Now I'm gonna try to do it. Oh, I think I lost my film. Okay, so here I have a film, I'm gonna try it. Oh yeah, it popped. Okay, so my observation agrees with what I predicted. Now, I want to try to figure out if I can do this, if I can do something, maybe to my finger, that would allow it to pass through the film. So maybe take a second to think about what might work. Okay, took some time. And again, pause this video if you want to take more time. I'm going to put my finger in the soap solution and see if that works. So I have my film. I'm going to put my soapy, oh yeah, see, look. I'm able to put my soapy finger right through the film. Now, if you're doing this, I should have given you some time to do it first before I showed you. But I want you to try to see if you can move your finger around, kind of like this, without breaking the film. That's pretty neat. Okay, so why can we do this? And how does this model getting something through a cell membrane? Okay, I'm gonna put this away for a second, but you're welcome to keep playing with it. And I'm gonna show you some pictures. I'm gonna have to turn my screen around. Here we go. Oh, that's not gonna work. Okay, so I'll just try to show you like this, except it's gonna be a bit backwards. So in a soap membrane, what we have soap is a molecule that has both parts of it that like water and parts of it that don't like water. So the way the soap molecules arrange themselves is they have the parts that like the water in the middle. You can see them, these like circles right here. These are called hydrophilic portions of the molecule, okay? You can kind of see that. And then they also have these tails here that are what we call hydrophobic, or they are scared of water, and they face away from the water. So what we've created here is a little layer or a bilayer of these soap molecules, where the hydrophilic part that likes the water is close to the center on either side, and the hydrophobic part is on the outside. And here we have the air and the air. So what we're imagining here is that this is one side of the soap film and this is the other. So when we put our finger that's just dry right through the soap film, these hydrophobic tails are scared of water, and so our dry finger doesn't really agree with them or the hydrophilic, the parts that like water part, and so the whole thing just breaks. But when we put our finger in soap, we get a bit of a different situation. So here, our coated finger now has a whole, this is simplified, but it has a whole bilayer of soap around it. And so these parts of the film here and here are able to kind of bend, and then you have the hydrophobic tails are now touching each other. So we have this kind of like touching like situation, and we don't actually break the film. And then when we remove our finger, these layers realign to look something like that. Okay, so that was a lot of me talking, less of you guys doing. So let's do a little bit more. So here again, we have our soap film. We've put a finger through it. That's not as complicated as it can get. What if we want to be able to pass something from one side of the membrane to the other that isn't coated in soap, or it doesn't have some sort of light component to the membrane itself? So to model this, I'm going to use another straw. So I have another straw in a different color. It doesn't have to be a different color. And I'm going to try to get this straw to go through the membrane. From what we know already, how do you think we can do that? Take a second to think about it. Okay, you can pause me again if you want to take more time to think. But I'm going to go ahead and guess that because when we put a dry finger through the film, it didn't work. And when we put a soapy finger through the film, it did. So I'm going to coat this straw in some soap and see if I can pass it through the film. I'm going to try to make sure you can see that. Okay, so I'm going to pass it from my close side to the far side. And look, it works. So I hope that you guys are trying this too and find it just as cool as I do. You can also move it around and it doesn't seem to be breaking the film. Oh, except I did. That wasn't very helpful. Okay, I think I'm going to touch the end. Now, because you have this whole system at home, you can try all sorts of different things here, and you might even want to do this with someone you're at home with and have them hold it while you play around. So now, what was our point? We wanted to see if we could get something from one side of the membrane to the other. So now we have this straw, and you'll see that there's this channel on the inside of the straw, right? It's a cylinder, and it's hollow. So we might be able to pass something through that cylinder. Now, I have a little I think this is chopstick. You could take a skewer or anything else that might fit into your straw. Do I still have a film? I do. I don't know if I can do this with one hand. I don't think I can. But you guys can imagine that we can put the soapy straw through our film, and then we might be able to pass something through it. Okay, so you guys can try that at home and see what happens, okay? Now, again, we're using, this system is cool just to play with, but the whole point was to try to understand something about a cell membrane, this complicated structure. So why would we care if we can put a finger through a soap film or another straw and pass something through a soap film? So I want you to take a minute and think about what you know already about cell membranes, or at least what we first discussed in this little video. And we thought about the function of a cell membrane, how it separates the outside of a cell from the inside of the cell, and how it's important to be able to pass things like nutrients or waste from one side of the cell to the other. So we might want to be able to create passageways through that membrane, or in this case, film. And that's exactly what we did. So the first little experiment was thinking about how to get just an object, like a finger, through the film. And we did that by coating it in a like material to the film. So this happens in cell membranes. There are certain molecules that are similar in their composition to the inside of a cell membrane, and if they're small enough, they can just pass right through. And this is called just simple diffusion across the membrane. You might be familiar with the term diffusion, and it just means that a material can travel from an area of high concentration to an area of low concentration without any added energy. A way you can visualize this is if you, say, have a bowl of water and you have some food coloring and you drop the food coloring in. So you can try that at home too. Or you don't even need food coloring. You just need something that has a different color to the clear water, and you can watch it dissipate. So when it first gets dropped in, it's high concentration, and then eventually it spreads out until you have an equal concentration. So if you have molecules that are outside of a cell, like oxygen, that are in high concentration, and the inside of the cell has a low concentration, that molecule is small enough that it can pass right through the cell membrane, which is great for the cell. Now, there are other molecules the cell needs, like glucose, to work, and those molecules are pretty big. They're also not the same kind of composition as the membrane itself. So to think about our model, they're not soapy going into a soapy film. So they would just break the film in our kind of analogy here. So we need another way to get them across the membrane. So I want you to think about what we did with our little extra straw, our channel. We put something that was like into the membrane, and we were able then to pass some information or a chopstick right through the channel. So I'll show you another picture let's look at a cell membrane. So we looked before at our soap membrane, and again, I apologize for the backwards writing. So we looked at this cell membrane, and we saw that we have the hydrophilic parts of the soap molecules, the soap membrane, on the inside, touching the water, and then we have these hydrophobic, water-fearing parts of the soap molecules touching the air. Okay, so think about that image, and then you can look at a cell membrane. So now we have the hydrophobic, the tails, the things that are scared of water on the inside, and we have the things that like water on the outside. And this is because the material that's on the outside of cells and on the inside of cells is water soluble. So these water-liking parts of the cell membrane, the phospholipids, need to be on the outside and on the inside of the cell. Okay, so what we just discussed is if molecules are like the inside parts of membranes, and small enough, they can pass right through. But if they're not like, they need some other mechanism of getting through. And so that's where proteins come in, and they embed themselves in this membrane and create little channels, just like our straw, to pass through the membrane. So now that you have a bit more of an understanding of how this model system that you've played around with can resemble or have similarities with a cell membrane, you potentially might have now, or I hope you have now, a bit of a better understanding of how things go into and out of a membrane. If you want to take this little exercise to the next level, you can take our other materials. We have our oil and our vinegar, or any other fluids you have, and you can dip your finger in those materials and make predictions of whether you think it'll go through our soap film or not. You might want to think about why one material might and why another material might not, and what these materials might be made of in relation to the soap, and you can make those predictions. You can also think of other ways to construct your little channel. Maybe it can let certain things through, like a chopstick, but maybe you can make this channel such that you couldn't allow other things through. And this could model molecules that maybe a cell doesn't want to be allowed to enter. Okay, so you could take this to a number of different levels based on your understanding or future studying of a cell membrane. I'm going to leave you with one thought, and that's that this little lesson was talking about using a model system, a soap film, a soap membrane, to model a complex system, a cell membrane. So again, this is something that happens often in science. It's important when we do these model systems, things that are really helpful, these simple systems that help us understand complex systems, to think about how they don't relate to the more complex system, or maybe some of their downsides. And that allows us as scientists to have a better understanding of the conclusions we draw from these simple models. Okay, so I'll leave you with that. I think what would be really helpful for you when you're trying to understand a cell membrane with this experiment, this activity, is to reflect on what aspects of it are like a cell membrane and what aspects maybe don't represent something that's happening in the cell membrane. Okay, so in the comments to this video, I'll leave a little link to areas you can read further to check your understanding. And the best part that I'll leave you with from this experiment is that you can blow bubbles. Okay, hope you learned lots. Take care.